In this video, I'm going to look at proteins and amino acids in general for a GOB audience, a general organic and biological chemistry audience. Okay, so proteins. Proteins are the most common molecule in biology. Roughly about 50% of your dry weight, so if you get rid of all the liquid inside you, approximately 50% of what's left would be protein. And proteins are made from amino acids. Let's look roughly what an amino acid is. So I'm looking at, let's see, let's use a red pen. Okay, so an amino acid here, they're also called alpha amino acids. And that's because we have an alpha carbon, which has a functional group which I'll call R. It has a hydrogen, and then it has two other things. It has a carboxylic acid end, and it has an amine end. Okay, so that's an alpha amino acid, and you can link alpha amino acids together to form proteins. Let's consider what that might look like. So let's just join two together. So I'm going to change my pen. Let's go to black. All right, if I were to add another alpha amino acid here, let's draw it real quick. That's lone pair, it's alpha carbon, its functional group. It could be the same functional group. Let's put a prime to mean it's different. Hydrogen, carboxylic acid, OH, the lone pairs on oxygen. And we know from our previous discussions in this sequence of videos that the lone pair can attack the carbonyl. The carbonyl, recall, is the carbon with a double bond to oxygen. So the nitrogen, or the lone pair of the nitrogen, can attack the double bond here. And there's some jostling around of electrons here, but ultimately we're going to lose this OH bond, which is going to snag one of these H's here. So essentially what's going to happen is we will form, and I'll go back to my red pen, we will form, I'll probably have to squish it down here because I'm running out of space, but H2N, the alpha carbon, R, H, the carbonyl over there. I'll try not to draw on a slant. I don't know why I'm drawing on a slant. Let's see, let's get rid of that funny C. Let's make it look like it really wants to be a C. Okay, that looks a bit better. And then we have that bond, so let's change the pen to black color. Then we have that bond. This bond, the arrow here is now going to be that black bond. And we've got a nitrogen. It retains one of its H's, and then it's got its alpha carbon with its different functional group here, NH, and then the carboxylic acid part. Okay, so we've And we could do this, etc., ad infinitum, as long as we wanted to. And I'll circle the key feature that we've formed here. So we're getting a lot out of this slide. This is an amide group. So an organic chemist would notice this is an amide group. And you can tell an amide group because it has a carbonyl, the C double bond O, and then a single bond to nitrogen. Okay, carbonyl, single bond to nitrogen. We could do that again. We could, continue the chain both sides. This nitrogen lone pair could attack a carbonyl on the left. This carbonyl on the right could be attacked by another nitrogen to its right. A biologist is much more likely, instead of calling an amide group, a biologist would probably call this a peptide group or a peptide, because peptides include amide groups, or you could call this a peptide bond, for example. So specifically this black bond here is a peptide bond. You could call it an amide bond. It's the bond within the amide group. Okay, so we got a lot out of that slide. So that's great. So let's move on. So at least for my students, I don't need, this is a chemistry class that I'm gearing towards for my own purposes. For my students, I don't need them to know examples of biological functions of enzymes. This slide for us is just to point out how useful and how important enzymes are in biology. So proteins can function as enzymes. Enzymes are essentially catalysts and they can accelerate biological functions. So I'll point this out at the top here. So literally just to be aware, enzymes are catalysts and catalysts can accelerate biological functions. Obviously, structure can be provided through proteins. We have, hair is an example of structure that's important in biology. We have other things like connective tissue and collagen and fibrous tissues that can all be important structures. Contraction of protein in the form of muscle, that's obviously important for mobility and for motion. Consider a world where you don't have any muscles. Not to mention, smooth muscle. You must have that, otherwise you won't be in the world. Transport, it can be used. We've just finished a video on lipid membrane. Proteins are involved. One of the processes for transport is transporting materials across the membrane lipid. Obviously that's important. Lipoproteins are involved in the formation of chylomicrons, which are the biological micelle used for transporting cholesterol and that type of material around the aqueous environment of the cell and the blood vessels. So obviously that's important. So transport, obviously key. The workers in the cell, a lot of work is done by proteins, making hormones and regulating the manufacture of hormones, etc., among many other things, etc. So again, the important thing about this slide, at least for my class, is just how diverse and how vital proteins are. And that's it. Okay. proteins and amino acids are the building block of proteins. So this is the, if you like, alpha amino acids are the brick, and you lay many bricks and you get a protein. Protein is also known as a polypeptide. And this would be the wall. So proteins are made from alpha amino acids. We've already, in the previous slide, the first slide I believe, we looked at the structure of these alpha amino acids. We talked about the amino group, that was the H2N part of the structure. And then the carboxyl group, that was the carboxylic acid part of the structure. Essential amino acids mean that they must be ingested in the diet. Why might some amino acids be synthesized in the body and others are essential? The reason for that is linked to evolutionary abundance. So for example, if a molecule was commonly found in the diet, there's less of a biological pressure to synthesize it. You know, if it's always available, there's definitely less of a biological pressure for the organism to synthesize it. Zvita ion, that's definitely an important vocab that we'll come to probably right now. So let's take a look at it. I don't know this for a fact, but my feeling is it comes from the German word for two. So I think the German word for the number 2, and forgive me if any fluent German people are looking at my my spelling here. I believe that's the German number for 2. So this is 2 in German, I believe. I'm going to put my neck out. Fewer and fewer people are willing to put the neck out these days, but I'm going to do it. I believe that's number 2 in German. So essentially, Zvita ion means 2 ions, right? And not just any 2 ions, but 2 opposite ions, a negative ion and a positive ion within the same structure. So for example, if you have within the same molecule, if you have something with a plus charge and something with a negative charge, then the whole thing has no charge. So it's important to notice that Zvita ions are electrically neutral, and that's because the sum of the positive and negative charges exactly equals and therefore cancels itself out. So Zvita ions are ionic to an extent that they become neutral, and that's an important point. Okay, let's have a look at, so the next couple of slides are going to list all 20 alpha amino acids, and we'll point out the Zvita ionic nature of them. So there are 20 alpha amino acids in nature. Essentially, for my class, anything on these slides would be provided to you in an assessment. There's some things that we'll point out, but you don't, for me, you don't need to memorize the structure and the name. You don't need to memorize the 3 or the 1 letter abbreviation. But I'll put the certain features that I do want you to know, so I'll comment on those as we go. So let's go through these. I'll try and make this a bit larger. Let's go through them. Okay, so glycine. Glycine is the first one here, and we can see that glycine, what I want you to know, glycine is hydrophobic. So what do we mean hydrophobic? Well, let's orient ourselves first. Here we have the carboxylic acid end. They've lost the H because they've written this in the Zwitter ionic form. Here's the amino end. This is the alpha carbon with its hydrogen, and then its functional group is hanging down here. In this case, the functional group is a hydrogen. So that's to orient you. We know it's a Zwitter ion, and these will all be Zwitter ions. These are all written in their Zwitter ionic form. And essentially, this is the form found physiologically. So when it's inside the host, the natural, if you will, form of these alpha amino acids is to be a Zwitter ion. So we've got a positive charge on the nitrogen because we've picked up an extra hydrogen. And we've got a negative charge on the oxygen because we've lost the acidic hydrogen. When I label this hydrophobic, I'm exclusively talking about the R group. Hydrogen is a low electronegativity element, and water, although water contains hydrogens, water doesn't really like hydrogen, doesn't really know what to do with it. The hydrogens in water, remember, can form intermolecular force bonds between oxygen and adjacent water molecules, but they don't really interact with fellow hydrogens. This hydrogen is not capable of hydrogen bonding because it's not on an oxygen or a nitrogen or a fluorine. It's just a run-of-the-mill boring hydrogen, and water has nothing to do with it. So it's hydrophobic. So I would like my students to know, looking at that structure, it's hydrophobic. Also, I would like my students to know that because it's got two hydrogens on the alpha carbon, it's achiral. So this is the only alpha amino acid that is achiral. So it is achiral, and that means it has 0 chirality centers. Okay. Check. All right, moving on to alanine. Alanine, the functional group here is a methyl group. Methyl is just a hydrocarbon, so it is also hydrophobic. Incidentally, hydrophobic just means water-fearing. I didn't mention it earlier because we've seen it in another video, but it means water-fearing. So this would mean that this alpha amino acid would not interact with water very well. It would be an oily blob on top. And that's going to be important later on. Okay, moving on. Oh, and before we do move on, sorry, getting ahead of the game. All right, let's point out to each other that we do have a chirality center for alanine, because the carbon here, again, in GOB chemistry, we're looking for a tetrahedral carbon with 4 different groups. So we've got an amino group, which is different than a hydrogen, which is different than a methyl, which is different than a carboxyl group. 4 different groups, so we can write this carbon with an asterisk. It is therefore chiral. And in fact, all remaining alpha amino acids are chiral at that alpha carbon. I won't label them from now on, but just make a note to yourself that you can see that it's a chiral center. And we should probably point out all natural alpha amino acids, obviously except glycine, are L alpha amino acids. Recall from our carbohydrate discussions that we would have D as the natural form of monosaccharides. D is dextrorotatory, it rotates plane polarized light clockwise. L, on the other hand, is levorotatory, and it would rotate plane polarized light counterclockwise. So L, recall, is levorotatory. Levorotatory. All right, moving on, valine. Valine is hydrophobic. Our functional group here just contains hydrocarbons, so hydrophobic valine. Moving down to leucine. Leucine is also hydrophobic. Again, we just have hydrocarbons, so hydrophobic. Again, the idea here is not to just memorize these labels, but rather, given the structure, can you use your chemical know-how to probe the functional group and decipher whether or not it's hydrophobic or not. Isoleucine is also hydrophobic. You can see it's an isomer of leucine. That's where the iso comes from. So it again is hydrophobic. Again, both chiral. So going down to phenylalanine. Phenyl or phenylalanine is also hydrophobic. Again, it's just we have an aromatic group here. This is a benzene ring, which, if you're not as familiar with the presentation given here, the skeletal structure, it has alternating single and double bonds. It's not an alkene, though, it's an aromatic. It's actually the only member of the aromatic family that we typically see in a GOB class. So it's an aromatic group. Essentially, it's just made of hydrogens and carbons, so it's hydrophobic. Okie dokie, moving on. We have tryptophan. Tryptophan is also hydrophobic. We look at the functional group here. We've got another benzene ring. Remember we said that nitrogen and oxygen really like water, but we said that you need at least 6 or more oxygens or nitrogens to, for want of a better word, drag carbons into solution, an aqueous solution. So let's have a quick look. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9 carbons and only 1 nitrogen. So clearly we have, sorry, I have no idea what I've just written there, but what I wrote was craziness. What I meant to write is not what I wrote. We need at least, let's see if I can go back to my pen. What I'm trying to say is that oxygen or nitrogen are capable of dragging approximately um, five carbons each. So if you have 6 carbons, you need 2 because that's more than 5. If you have 10 carbons, you need 2 and you'll just about do it. If you have 11 carbons, you need 3. So what am I trying to say? I need at least, yeah, I'm actually going to rewrite that because I can't even salvage that sentence. That's how bad it was. We'll get there. We will get there. Okay, so let me clarify for the sake of science. Okay. Each nitrogen or oxygen is capable of dragging about 5 carbons into solution. That's what I'm trying to say. So here we just have 1 nitrogen, but we had 1, 2, 3, 4, 5, 6, 7, 8, 9. So here we have 9 carbons, but only 1 nitrogen. 9 carbons are going to win, so it's hydrophobic. That was painful, but we got there. Thanks for sticking with it. Moving on, methionine. Methionine is hydrophobic. Hydrophobic, we just have hydrocarbons. We have sulfur here. Sulfur is a little bit exotic, but it's roughly the same strength as a carbon. So there's not much here that has any interest for a water molecule. So the whole thing is hydrophobic. Okay, let's see. Going on to prolene. Prolene is also hydrophobic. It's a cyclic functional group here. In fact, let's break this down a little bit. You can see here that this is the alpha carbon here. This obviously is the amine. So what's happening is this really, the functional group, if I can tease it out a bit better than, to be honest, I have done. Let's try and tease that functional group out a bit. Okay, the functional group is just these carbons here, because we would include this nitrogen as part of the alpha amino acid backbone. Okay, so we have hydrophobic proline. Okay, moving on to serine. Serine now is our first example of a hydrophilic functional group. Hydrophilic. Hydrophilic means water-loving. Means it will dissolve with water, interact favorably with water. We've got 1 oxygen, only 1 carbon. The carbon's no match for the oxygen. We can hydrogen bond with water. We can dipole-dipole bond with water. We can London dispersion, interact with water, etc. Threonine. Threonine is also hydrophilic. Functional group here. Hydrophilic. I'll try and go through these a bit faster because they're kind of repetitive. Cysteine is hydrophobic. Let's mix it up a bit. Hydrophobic. Again, sulfur is about the same electronegativity as a carbon. So it's like a fuzzy carbon in a way. So it just looks like an exotic hydrocarbon, hydrophilic. Tyrosine, we have hydrophobic. Big aromatic ring there, only one oxygen. Asparagine is hydrophilic. Hydrophilic. Again, we've got two carbons, but we've also got a nitrogen and an oxygen to overpower them. Glutamine. Glutamine is hydrophilic. Hydrophilic. Again, we've got a couple of carbons here, but we've also got a couple of, well, an oxygen and a nitrogen that can overpower it. Probably getting close to ending this list. Aspartic acid. Aspartic acid, or you could say aspartate, I guess, is hydrophilic. Okay. We have glutamic acid or glutamine. No, sorry, glutamic acid is, what is glutamic acid? Glutamic acid is glutamic acid. Guess you could say glutamate. Hydrophilic. All right. Histidine. Histidine is hydrophilic. Hydrophilic. Try and get through this list. It's kind of repetitive, but it's important. We'll only do it once. We won't actually repeat it. Hydrophilic again. And then once you have this list, you can practice it and, again, not memorize, but try and interpret the structures. Arginine is hydrophilic. Again, look at all those nitrogens there. More nitrogens than you know what to do with. Okay, so that does all 20 natural alpha amino acids. Great, so moving on. We've already, I guess in, what was it, slide 1, I think we just went for it in slide 1, so just to refer you back to this first slide, we saw that alpha amino acids can interact with each other to form long chains. So go into the current slide, it just formalizes that with some words. So alpha amino acids can attack neighboring alpha amino acids through acid-base chemistry. So that's really important. So they exhibit acid-base chemistry. And therefore they are sensitive to the pH of their environment. This has connotations for health. This also has connotations for food and pickling and preserving food, and there's a lot of history here that we sadly probably won't have time to go into, but it's interesting nonetheless. So let's see. Okay, so let's have a look. So it can react as both an acid and a base. The base part is from the amino part, the NH2 part with the lone pair. And the acid obviously is from the carboxylic acid. The C double bond OH part. Okay, we've already talked about Zwitterions. We know that naturally the amino part would be positively charged. The carboxylic acid part would, in the Zwitter-ionic form, be negatively charged. And this slide is just reminding us of that. The isoelectric point. The isoelectric point, as said here, is the pH at which amino acids exist in a solution as the Zwitter ion. The isoelectric point is often abbreviated Pi. So typically, here's a great question that I typically ask my students. Under acidic conditions, under neutral conditions, or under basic conditions, what is the structure of an alpha amino acid? Well, the neutral, let's have a look. We would have our alpha carbon with its payload of a functional group. I'm going to ignore the lone pairs on oxygen. We know that they're there. I don't have to remind us each time. So under neutral conditions, you would have the Zwitter ionic form. This is at the Pi, which essentially means physiological pH. So the pH that the protein finds itself intended to be in the host. And that's that. What if you were to take the alpha amino acid outside of the host and you were to deliberately add acid? So this would be to go to a pH lower than the Pi. Well, we know from the Brensted-Lowry definition of an acid, it's a proton donor. So if we took the Zwitter ionic form and we just peppered it with protons, we would quench the negative charge here and we would leave the amino part unaffected. So let's have a look what that would be. That would be H3N as the alpha carbon, R, there's the H. We would quench this, get rid of, so it's no longer a Zwitter ion, and as you can see, it's a net cation. So it's a cation, and that means it's positively charged. The Zwitter ion, remember, means it's neutral, or no charge. And then if we were to put this alpha amino acid in a basic environment, where the pH is now greater than the isoelectric point, we would essentially remove a Brensted-Lowry base as a proton acceptor, so we would accept protons. They have to be acidic protons, so we wouldn't accept this H, for example, or any H's down here necessarily. We would accept this H. So that would give us only 2 H's. And therefore we would lose that positive charge. and we would be left with an anionic form of the alpha amino acid, which is a negatively charged ion. So this is something I typically ask my students, and I would like them to pay close attention: the effect of pH on the structural presentation of alpha amino acids. Okay, moving on. And we'll probably pause this video around slide 18, and then we'll conclude the video with we'll conclude the slides with a second video. The isoelectric points of some representative alpha amino acids. Again, I don't need anybody to, or I certainly wouldn't suggest that anybody memorize these things. So these isoelectric points, the only thing I would point out here is if you were to average all of these, they, on average, they're approximately pH 7. Obviously, they're not all 7, but they tend to hover away from the extreme pH ranges. So there's no 14s here, and there's no 0s and 1s. So they're all approximately 7 or thereabouts. Okay, that's the only thing I would say is of relevance there. So let's have a look at this. We've mentioned the fact that peptides, or at least we mentioned that the peptide bond is also known as an amide bond. And we know that to a chemist, which is what we are, is going to be that bond there. So it's when you have a carbonyl and an amino group adjacent to each other. That's an amide bond. It's also called a peptide bond. And it's the result of when two alpha amino acids come together. And just to remind you of that, we've written that here. Maybe I've never tried to copy anything before, but let's try it. You don't try, you never know, do you? So I'm going to try copy and paste just to put it in this part of the slides. Let's move this out the way first. Making a mess of this, aren't I? Let's move that up here. Let's maybe move this again. I really want that to show. Let's move that down. How about that? And then we'll try and get that copied here. Maybe if I can group it, and then I can maybe make it a bit smaller. Let's get that up there. And then maybe I can get this back on the thing again. Oh, perfect. Okay. Okay, so we've seen a fact what the amide bond or the peptide bond is, and just to repeat from the first slide how it might actually form. One thing I didn't point out, I guess, in slide 1 is notice we lost this OH and an H. So just in the pure excitement of forming the amide bond, I guess I never mentioned that you also form a water molecule. Water is not that important here because, I mean, everything's water, right? There's so much water in biology. How are you going to notice that water molecule? But there's water there as well. Okay. I think this, let's see, this might be our last slide. Yeah, I think this would be a great place to finish the first video, actually where we were here. We'll finish here, and then we're about halfway through these slides that I wanted to present to my class. So we'll conclude it here, and then I'll make a second video where we will conclude our discussion on proteins.